most of us all of us studied Mendel's work and his contribution right from our school days. And it's very important finding which was recognized much later. But there are several scientists followed his work and reinvented and subsequently agreed upon that whatever he reported was in fact correct. And even today, we're all following so whatever he invented about maybe 150 years back. Although it appears that it's kind of modern science, but in fact, some of the common people also knows about genetics, right? Even in the remote village, if you go, if you ask if there is any disease, then you say that oh, it was there few generations back, or it was there last generation, and it has come to this generation also. And even if you go to farmers, they may say that so they are looking for a better variety, right? So any field if you take, there's always a genetic. Not only the scientists or educated people, even the common people knows about the kind of inheritance. That's a major concept of inheritance of genes and the chromosomes. Therefore, it is very important for us to celebrate his 200th birth anniversary in a way to recollect his finding and to look at how we are now in a position to adopt his finding and how we are making our own conclusion using genetics or genomics, particularly for mankind. I hope all of you like my talk. That my talk is basically on genetics, human genetics. I'll start from human origin, then try to understand how understanding human origin will help in understanding a disease. Then you can clearly see that what Mendel found about 150 or 160 years back, that how the either you can say DNA or chromosomes are inherited, and the dominant character, recessive characters, all these you will find in my talk. I'll highlight some of those. I'll share my slide. As I mentioned that I'm going to talk about mostly the human genetics, and before that, shall I give my tribute to Mendel for whatever he has made, his contribution, and because of his contribution, the world is celebrating his 200th birth anniversary. And his observation we all know, but what was interesting to know is that he presented his work in one of the annual meetings of Society for Study of Natural Science in the year 1865, but no one asked anything because probably no one understood that. Then he also published in the local journal. Not much reference was made for this particular paper in several years until the three scientists, Hugo de Vries, von Chefmark, and Carl Carens, who have independently rediscovered his finding and the principle of heredity. It was not well known to everybody. So we must thank these three scientists also for bringing his principles of heredity into the light of many people or scientists. So in that sense, that as I mentioned earlier, that these findings are very important. Until today, the common people are well aware of the hereditary aspect. Another scientist who made a significant contribution was Charles Darwin, who was almost a contemporary of Mendel, and both of them contributed very, very significantly to biology. So my talk, fortunately, that my basic research for the last 30 years is based on both of their principles. That's what I'm going to present to you today. I've been working on human genetics, two different aspects. One is understand the population history using genetic as a tool, again, Mendel's concept. Then the second aspect is understand the genetic aspects of various diseases, some of which I'm going to touch upon in my talk. If you wanted to understand the population history or look at the population as a whole, there is no other country than in India or South Asia, because we have huge amount of diversity in terms of our color, in terms of our culture, in terms of food habit, and many more. Some of the population where you see, they typically look like Africans. Some of the populations are typically with very light skin color like European. Some has the features of Southeast Asia. And some of the populations are very unique to India. So such a huge, complex country. And the Indian populations are very well stratified in many ways. One of the stratification is based on the language, because it's animation, so you could not see the previous slides. It is there behind this. So we have four major linguistic groups in India. One is Dravidians. All the 5 states speak Dravidian language, like Tamil, Malayalam, Kannada, and Telugu. Similarly, we have 3 more linguistic groups: Austroasiatic, Tibeto-Burman, and Indo-European. Again, like Dravidian, each one of these also has several language groups. Then under each linguistic group, there are several social groups, like we know that so-called upper caste, middle caste, lower caste, tribes, and primitive tribes. We also have religious groups. So all of this makes India as one of the very complex country. To understand the complexity of the origin of population in India, we have to look back kind of how the human population originated and how they migrated and how the Indian population originated. You have to understand little more about the origin of modern human. Origin of modern human, we all know that was in Africa about 160 to 200,000 years back. And from there, they migrated to any part of the world. How did they migrate? There are 2 hypotheses. One is that a group of scientists, historian, linguists, anthropologists, archaeologists, they all believe the first modern human migrated out of Africa taking northern land route. Others don't believe on that because the climatic condition in Europe probably several thousand years back might not have helped people to migrate through that. Whereas the others, they have believed that they must have taken the southern coastal route. They're all based on the archaeological evidence present in these places. Archaeological evidence suggests that modern human arrived Australia about 50,000 years back, China 39,000 years back, Middle East 47,000 years back, and Europe 40,000 years back. But unfortunately, we don't have any good archaeological tool to suggest that when did the modern human arrived in Indian subcontinent. There are few very recent studies which I did not keep here that suggests that the samples are not very old. One of the study from Harappan region that was about 3,000 to 4,000 BC old. So naturally, that is not very old sample to understand when did the modern human arrived. In the absence of good archaeological samples, our idea was looking at the contemporary population's genome to understand the early human migration. We used different genetic markers. One from the nuclear genome, which we all inherit equally from both the parents. We have 46 chromosomes, 23 we inherit from the father, 23 from the mother in the form of sperm and egg. These are equally received from both the parents. So among these, there's one tiny chromosome called Y chromosome that is present only in the male. So all the males have in the same family will have a similar Y chromosome sequence because it inherits from grandfather to father, father to son, then grandson without much change. So if you want to trace the paternal lineage, we use the Y chromosome marker. Other one is mitochondrial genome. This is called as non-Mendelian inheritance. We inherit the mitochondria and mitochondrial genome only from the mother. Right? That's in the egg, the mitochondria will be there, but in the sperm, mitochondria will be in the midpiece of the sperm. Therefore, it remains outside at the time of fertilization. Therefore, we don't get father's mitochondria. We get only the mother's mitochondria. In a family, all the children will get mother's mitochondria and mitochondrial DNA. The son cannot transmit to the next generation, whereas the daughters can transmit to the next generation. So it is going through maternal lineage. If you wanted to trace the maternal lineage, use the mitochondrial DNA. If you want to trace paternal lineage, we use Y chromosome. If you want to look at both, then we use other nuclear genome. I'm going to give you a very small or very significant findings by looking at the mitochondrial DNA and also the nuclear genome. So one of the important hypotheses was that the modern human came to India from Africa through southern coastal route. So we wanted to establish that. Therefore, we collected the sample from the Andaman tribes. There are 4 tribal population who typically look like Africans. They are Great Andamanis, Jarwas, Sentinelese, and Onge. 2 of them typically look like Southeast Asians. They are Nicobaris and Champagne. Their number is, all of these are very, very less except the Nicobaris. So we collected the samples from all the group except Sentinelese because they are in isolation. Anybody goes near them, they will shoot with the arrow. So we collected the sample, then we sequenced the complete mitochondrial DNA, which is of 16,500 base pair. Based on the mutations, one can assign a particular haplogroup to every individual's mitochondrial DNA. And this is called mitochondrial DNA parsimonious tree. And you can see the distribution of the haplogroup in all over the world. You can also see some haplogroups are restricted to only in Africa. These are L-derived haplogroups called as, this is the root of the mitochondrial haplogroup. In addition to that, these are all defined by a set of mutation, which I'll show in the next slide. In addition to these mutations, there is another set of mutation. The individual's mitochondrial DNA sequence fall into M haplogroup or in N. And every set of mutation, these are evolutionarily conserved mutation, can help us in putting a specific or assigning a specific haplogroup. When we sequence the complete mitochondrial DNA, we found the Onges and Jarvas are Great Andamanis are having a specific set of mutation. The number, what you see, 200, 1,524, these are the nucleotide position. I said 16,500 base pair. In that, there's a mutation at the position of 200, a mutation at 1,524, like that. These mutations are not found anywhere in the world. These are very unique to Andaman tribes. And based on the mutations, one can also calculate age of the population. So based on that, we have concluded that the Andaman tribes are the first modern human migrated out of Africa, taking southern coastal route and reached Andaman and Nicobar Islands about 65,000 years back. So this is a very first genetic evidence suggesting that the Andaman tribes are the first modern human who migrated out of Africa. And from there, they migrated to Australia, and several studies confirmed that Australian Aboriginals, Andamanis, and Kurumba from Kerala, all having some kind of genetic affinities, confirming our findings that the first modern human migrated out of Africa, taking southern coastal route. After that, there are several population we have studied, but I'm going to tell you only the significant findings. Then we studied large number of population using the nuclear genome. I said that nuclear genome, except the Y chromosome and also the X chromosome, equally comes from both the parents. After human genome was sequenced, the nucleotide, which shows the variation between different ethnic groups, have been identified. Now 1 million genetic variations can be analyzed in a single experiment. They're all spotted in a single glass slide. So we selected a population based on our previous study, population representing all the 4 linguistic groups: Dravidians, Indo-European, Austroasiatic, Tibeto-Burman, and also Andamanis. And we also carefully chosen the representative sample from so-called upper caste, middle caste, lower caste tribes, and primitive tribes. Okay. And we used the already existing data from Human Genome Diversity Panel and HapMap data. These are from different continents, which I'll tell you in the coming slides. We use several statistical tools to understand the Indian population history. Our findings based on this study gave very different clue that that helped us in reconstructing the Indian population history. So what we found was that this is the PCA, principal component analysis. It shows that this slide alone tells that how complex Indian populations are. It tells that India is not inhabited by a single large population as we see in Europeans, Africans, and Chinese. There are many samples in these clusters falling in a single type cluster, suggesting that genetically they are more homogeneous, not much different, therefore everybody is in a single cluster. Whereas look at Indians, every single population is unique. Although every population is unique, but there are large number of genetic markers which are common between some of the populations. That's why there is a cluster after the cluster, which are, we can say that gradient of clusters, suggesting that populations are very unique, but still there's some genetic association between the group. We came to know later on why this kind of affinities, or how this kind of affinities. So based on several other statistical analysis, we came to know that all the Indians have admixed. There is no single population in India without mixing. Everybody is mixing from 2 founding groups. One is ancestral South Indians. They are part of the early human migration. That means when Andamanis were migrated from Africa, that's a deep-rooted divergence. Some groups stayed either in Kerala or Tamil Nadu, whom we call as ancestral South Indian. There was another group which migrated from Africa towards north. One group has gone to Europe, other group has gone to northern part of India, whom we call as ancestral North Indians. And these two groups have admixed. When this admixture took place, we took another study which will come a bit later. These two groups have admixed and gave rise to many population groups. And why they are saying that the Europeans and ancestral North Indian has some kind of affinities? There are several evidence. The mutation which made the skin color of the people into lighter skin color, that mutation is common in Europe, Middle East and northern part of India, but not in southern part of India. The Y chromosome haplogroup R1A, again common in all these groups, Europeans, Middle Eastern and ancestral North Indians. The lactate persistent gene mutation also common there. So all this suggesting, we all confirmed with large number of studies, suggesting that there's a kind of affinities. These are the major findings. Only that all Indians have admixed, because this is what we are saying, there's no population in India without any mixing. And we also refuted Aryan invasion theory. So you know that there's a theory proposed by Max Muller that the light skin color people are Aryans. They pushed the dark skin color people towards south, we call as Dravidian. That means the Aryans pushed the Dravidian towards south. That we negated that there is no Aryan invasion, because we didn't see any distinct population in India as a distinct cluster suggesting that they are Aryan. Everybody is now mixed. We also predicted high frequency of recessive mutation, recessive disease in India. In addition to these two founding groups, there are several groups which have migrated recently, like Alexander's army, military conquest of Arab and Turks, British colonization. There are several groups migrated very recently, like Siddhis, Jewish, Parsis. We have studied all of these populations. Because of time limit, I'm not going to tell. But what I wanted to highlight is, because of several groups which come very recently, and the founding group, ANI and ASI, it makes India is one of the very complex nations. And it's not exaggeration to say India is an ethnic group, because we have collection of large number of population groups. Like we have 4,635 human population groups. Nowhere in the world you will find such a huge diversity. 532 are tribes, 72 are primitive tribes, and half of them still practice hunter and gatherers lifestyle. That means they just go and hunt for food. They don't do any job, including agriculture. So what I am going to tell you now is that I mentioned earlier that there was admixture between ancestral South Indian and ancestral North Indians. That time, we don't know when it happened, but in the subsequent study, which we took using large number of population from South, large number of population from the North, then we came to know that these two groups, ancestral South Indian and ancestral North Indians, individually gave rise to many population groups, right? Until last 4,000 years. That means the population settled in jungle, and some group must have migrated from the deep forest into maybe another place, and from there some group must have migrated to somewhere plateau region where they started agriculture, like that, many groups. Then these two founding groups have admixed during the last 2,000 to 4,000 years. The color indicates that the genome of these two with a different proportion because of the admixture. And what is interesting is that after this admixture, which happened till last 2,000 years back, and every single population started maintaining the endogamy, or means marrying within the group, either caste or tribe. So this came much before this attitude, or this aspect came much before the caste system. Probably the caste system, which arose around 1,500 years, must have put more pressure on every population. Okay, you now marry within your community, right? So that is have been happening for the last 2,000 years. But now what I'm going to tell you is what is the impact of this endogamy, marrying within the group. To understand that, we took another study. This time we have not restricted to only India, but we wanted to look at other South Asian countries also. Because again, this decision was based on our previous studies, which I'm not showing here. One of the study where we looked at South Asians, and half of the genome from India has gone to other South Asian countries. Okay, genetically, that means whatever the genetic mutation we find, at least 50% of that you may find in other South Asian countries. And some disease mutations, particularly the myosin binding protein C3 mutation, which we found very predominantly in India, was also found the same frequency in South Asia, but not in other countries. So these are the things which we felt that we should do this study for South Asians. We took 2,800 individuals from 275 endogamous groups, and the color indicates different linguistic groups. We also use the isolated population or the isolated language like Andamanis, Nihalis, and a few more isolated groups. And what we found in this study was that there are certain mutations which are very, very unique to every single population. And probably those mutations are responsible for causing a population-specific recessive disease. So that gives a lot more opportunity for the next generation students who wanted to study genetics to pick up all of this information and map the gene, study it thoroughly, and try to help the population in general, because 1/3 of the population in India are likely to have this condition, population-specific recessive disease. So the reason why we are saying is, so this is called as IBD score, identity by descent. Identity by descent means, in a simple term, if you take two unrelated individuals and if you compare their sequence, if they are really unrelated, then you will find variation between the individuals. But if they say that they are unrelated, but the sequence of both the individuals are identical or very less variation, that tells that today they are unrelated, but few generations back they must have come from a common ancestor or common parent. That you might see in the village, there are 50 families or 100 families, and they are marrying within the group. They all 100 must have come from a single parent. Maybe some 50 generation back, there was one family settled there, and that family has 4 or 5 children. Then they expanded like that. So in that scenario, what happens? After a few generations, maybe 20 or 25 generation, then now they may say that they are unrelated, but actually they have come from a common ancestor. That means if there is a mutation, the founder, so that is going to be in both the families, and they are, or you can say that both boy and girl from those two families. And if it happens to be a recessive mutation, the recessive mutation, there are two broad kind of mutation. One is a dominant, other one is recessive mutation. If there is a dominant mutation, because I mentioned earlier, we have 23 pair of chromosomes. 23 we inherit from the father, 23 from the mother. So every chromosome take, there's a pair. If one of the chromosome of the pair carry the mutation, if it expresses the disease, that's the dominant. No problem. Whoever having the mutation, they're going to have the disease. But the problem with the recessive mutation, if the mutation is recessive nature, one chromosome carries the mutation, other one is normal, and the person will be absolutely fine. No problem at all. Because every population marrying within the population, therefore that the chance of increasing of the frequency of that particular mutation in this scenario is quite high, which I'll tell you using the pedigree. So that observation we made by looking at different population groups. Every line representing one population. And I mentioned here two population with a red line. One is Ashkenazi Jewish, other one is Finnish population. These two populations are small size, also like they practice endogamy like Indians. And there are several diseases have been mapped in those populations. Where you see these are the diseases, these are the genes responsible for disease. These are telling that most of them are autosomal recessive. Okay. That happens because of the endogamy, small population. Similarly, Finnish population, large number of diseases. Even with the small IBD score, the length of the IBD suggests that what is the proportion of the population carrying the recessive mutation. That makes much more significant way that they will have a population specific disease. Even with this tiny IBD, you have seen large number of diseases, but you can imagine what is the impact of large number of population. They have several fold IBD compared to these groups. So to understand that, this is the pedigree. You can see most of you might be aware of what is meant by circle. That is the female, square is the male. If you put the line here, that means they are wife and husband, they're married. And if you put line below and you put circle, that means the daughter. And if you put square, son, and all of these are their offspring. Okay. In this family, two daughters, and this is a chromosome carrying the mutation in a heterozygous condition, right? This chromosome is fine. This male is fine. Then she is transmitting that chromosome to both the daughters. She married somebody from outside. She also married from somebody outside. Both of them are not carrying the mutation. Therefore, this mutation goes as such without any problem. Now, in this generation, or you can also take it to several generations. Now, this boy carry the mutation. This girl also carry the mutation. Now, if these two marries, and there's a statistical significance of 25% of their children might carry this disease. So you can correlate with Mendel's law, 1:2:1. Okay, so one opportunity of carrying this, that's a 25%. In one, there are a couple of 50% are heterozygous, again 25% are wild type. Okay, so to understand this in much better way, we took one known autosomal recessive disease. It's called as progressive pseudorheumatoid dysplasia. We took this patient, and this is caused by WSP3 gene mutation. This is a well-known recessive disease. Parents are absolutely fine. We took some 30 patients. Parents are absolutely fine. When we sequence, all the patients were carrying the mutation in a homozygous condition. And you can see it's a swelling of all the joints, and it's very painful. Now we wanted to fit with our hypothesis that most of the population practice endogamy, therefore this mutation comes from a common ancestor. We sequenced and we found 6 patients carrying the mutation, one particular mutation. And when we sequence the flanking region of the mutation on both the sides, and we found 5 out of the 6 patients who carry the mutations are having the same sequence. That means today they are unrelated. Probably several generations back, they are related. This mutation must have come from a common ancestor. Okay. So this is very well established. Subsequently, we found many other diseases. So this is another example. This is a kind of one of the mitochondrial disorders. I must say that mitochondria, initially told, inherited from the mother, and it has been thought until very recently, all the mitochondrial disorders are maternally inherited, doesn't follow a Mendelian inheritance. But in the recent past, when we sequence the mitochondrial DNA, most of the patient doesn't have the mutation in the mitochondrial DNA. Then we try to understand by looking at the nuclear genome. In fact, there are large number of nuclear genes which control several activities of the mitochondrial function. One of the activities is mitochondrial DNA replication. For that, the nuclear genome, which codes for the enzyme polymerase, which is essential for mitochondrial DNA replication. So that directly correlates that the mitochondria require nuclear genes for the mitochondrial DNA replication. So in this case, there's a patient, young children with the severe encephalopathy, seizures, stroke-like episode. And most importantly, they're dying at very, very early age. 13 years and 10 years, here 9 months, 18 months, 18 years, or maybe 18 months. This is in the consanguineous family. This clearly shows that there's a inheritance, recessive disease, which goes to the next generation. This is a sequence, shows this is a wild type, C. This is a complete mutant, T. So in this case, one chromosome is carrying C, other chromosome is carrying T. That means this is the heterozygous. The parents are heterozygous in the consanguineous family. This mutation must have arose somewhere in the family. Now, they are absolutely fine, 45 years, 39 years. They have passed the abnormal chromosomes to the next generation. Right? 3 out of 2 are carrying the mutation homozygous condition, which leads to several abnormalities. It's what you see here because of the mutation, single base mutation, which leads to multiple mitochondrial DNA deletion. And these are COX negative fiber. This is the blue ragged fiber where the abnormal mitochondria are accumulated. So what I am trying to say here is for a very long time, the mitochondrial diseases are considered as a non-Mendelian disease. Now we are finding that they also follow the Mendelian pattern of inheritance because it comes from the mutation comes from both the parents. In the consanguineous family, in the non-consanguineous family, also it appears like this. So like that, there are several such disease, and there are several young kids are dying because of these lethal mutations. And these are not even recorded in many cases are not recorded in the medical community or either ICMR. And most of them are in the rural area and it's not going to go to the medical community at all. So it's up to us to identify this kind of patients or the family and do the genetic analysis and try to help the family. So this is another example. A small village near Madurai in Tamil Nadu, there are large number of such cases. Junctional earlitz epidermolysis bullosa. It causes skin rashes and congenital heart disease. When we sequence the exome of several families, we found 11 base pair deletion in the LAMB gene. So that means it requires huge amount of studies in the population, which we are now collecting sample from most of the families, and we wanted to screen the entire population because most of the populations are individuals are carrying the mutation heterozygous condition. It will actually help in the next generation. We can actually give pre-marital counseling. And once you identify the mutation, you can see that whom to marry, whom not to marry. In fact, that's happening in Ashkenazi Jewish. They sequence the genome of students who goes to the school. They collect and the sequence and keep it in the database. when they grow and ready to marry, and they in fact identify the mutation in if they identify the mutation in a boy, they will check whether the same mutation also exists in the girl. If that is the case, then they will give appropriate counseling, either not to marry, or you can keep an eye on this mutation, and you have to do the prenatal diagnosis in order to prevent the disease. So they have been practicing very effectively. In fact, by looking like that, they eliminated a couple of diseases. So that is very, very important, and we need to do that. So what we are doing now is that we have selected the population with very high IBD score. We also selected the population with known population-specific disease like Handikudu, Udanam, kidney disease, and this is Vaishya population known for the soda-colon esterase deficiency, which because of that it leads to paralysis if regular anesthesia is given to this population. And what you see here, this is the Handikudu disease. The people are very short-statured and bone abnormalities and with very severe pain. And here what you see that is that Udanam kidney disease, where most of the people are undergoing dialysis. So now our priority in research is taking clue from our earlier study, IBD score, and also collecting sample from the known population-specific disease, sequence that, identify the mutation, then go for giving counseling to the population. Through this, we can able to help the community. And I must say before concluding that because of the concept of Mendel, that we could able to achieve this much, and we could able to help the people, common people, and the common people knows about the value of such research. With that, I conclude by thanking all of those who have helped in this study. Large number of collaboration and funding has come from CSIR, UKRI, DBT, PST, and MRC, London. That I thank all of you for listening to the talk.